Let's see. Okay, so the exam solutions have been posted, and I finished grading. So I just now finished emailing back everybody's graded exams. So to your whatever the email address is you have on eCampus, whatever your official A&M email address is, that's what I sent your annotated exam back to. After a curve, the grades went from 80 to 100, and for the most part, I was happy with how things went. There was one or two things I wanted to touch on. The data that you played with, let's talk about it for a second. Okay, so we have omics data where there are 8 assays. So we have a matrix of numbers with 8 columns. And there are 2 conditions that are going to be compared. And there are 2 cell types that are going to be compared. So basically this is a human population, and we obtain cell samples of a particular type from people. Right. So let's look at the data. Let me get a text file. So let's read in the data. And you could use this command right here to do it. So data is s.matrix to make sure I am not working with a data frame. Exam1.csv. Okay, so we have 54,000 genes. Here's the first 6 rows, and we can see the structure of the data by looking at the column names. In particular, the first column is cell type Th17. It's a control sample, so control condition, and this is donor 1. So you were told in the problem that there's only 2 people. We have 8 columns, but there's only 2 people that are giving us the genetic material to look at. First 4 columns are all the same donor, 1 person. So we have Th17 cells from this donor, and some of them were subjected to the control treatment or condition, some were subjected to the treatment condition. And then the last 4 are for donor 2. Okay. All right, everybody did well on the clustering. So when you did clustering, it was actually kind of a nice story that came out. So to do clustering, we can say hclust. I want a distance matrix. And then I think I need to transpose data. That's like what we did in our homeworks. Method equals complete. And we can plot that, and we can plot the merge heights. So this was what came out of hclust. And looking at the merge heights, how many clusters would you say there are? It's not a perfectly easy answer to come up with, because definitely looks like there was a substantial jump here. Maybe there was a substantial jump here, maybe there was one there. I didn't take points off if you said 4 and I said 2, as long as you kind of argued for why I think there's 4 or why I think there's 2. I'm happy with it because we've admitted that clustering is going to be somewhat subjective every time. My answer was 2. I tend to want to be conservative in interpreting groupings, because as we've seen, you will get groupings even when there aren't groupings. I mean, the cluster algorithm is going to tell you here's a dendrogram even if there is no clustering going on. So I guess I recommend being hesitant to over-interpret these pictures. I feel comfortable saying that there is a jump. I feel even more comfortable when I look at the dendrogram and it looks believable that there are 2 groups. These look distinct to each other, from each other. And even more comfortable when I notice that there's a sensible explanation for this grouping. So if I say there's 2 clusters, that means these columns went together and those columns went together. And what differentiates those groups is the cell type. So these are all the Treg cells, and these are all the Th17 cells. You can also see at a more subtle level, one that I wouldn't feel as comfortable really emphasizing, that here is donor 1 Treg cells, and here is donor 2 Treg cells. Here's donor 1 Th17 cells, here's donor 2 Th17 cells. So it looks like the biggest source of structure in the data is cell type. Next, it looks like the biggest source of structure is donor. And of interest is that none of this clustering seems to say anything about control versus treatment. We don't see any evidence of differences or distinctions based on control versus treatment. Treatment didn't cluster together and control cluster together, for example. Okay, and then part B was where I wanted to make a comment. So do principal component analysis to explore the columns. Should you standardize or not? As long as you justified what you said, I was happy, but in this case, you definitely did not have to standardize because the scales were already comparable. The column, the standardization versus not standardization question, remember, is one of, are the things we're about to search for structure among, in this case the columns, do they differ in scale substantially? So like, do we have one column that has a variance of 1 and then there's another column that has a variance of 100? If we did, we would definitely want to standardize, or all we're going to get out of PCA is evidence that one variable is super variable compared to the other ones. It didn't need any fancy matrix result to learn that about the data. So that's not really that's not what I want to do with PCA. In this case, many of you looked at side-by-side box plots, and you could see from them that the arrays, the columns, were lined up very nicely. They probably have already been normalized somehow, so it did not make a difference. If you did anyway, or no, we were all supposed to not standardize, right? Okay, everybody got that fine. No problem. Part 4, I think there was only 3 people who got this one right. So I wanted to address it. The question is, provide a scatter plot of the first two principal components. So when we do principal components, let's do that. This was clustering. And here comes PCA. Okay, when we do PCA, we can use the PRComp function. So I could say PCA is PRComp data. When you just use PRComp, the default is to not standardize your data prior to using it. And that's equivalent to doing eigenvalues, eigenvectors of the sample covariance matrix. So sample covariance matrix is this thing. Here's the eigen stuff for that. Let's do that and then verify that we're doing what we think we are. So PCA looks like that. These are the eigenvectors of the covariance matrix. So if I do eigen S, first column here, the first eigenvector is the same as this column here, except that this one has been, the sign has been negated. Negative versus positive does not affect the interpretation. This is saying first PC is basically an average. This is saying the exact same thing. The negative does nothing in terms of interpreting what these mean. Okay, so that's all we've done with PRComp is this decomposition of the covariance matrix. Okay, and then provide a scatter plot. All right. So what are the principal components is the question that I want to clarify. You are asked to provide a plot of the two principal components, first two principal components. And the principal components are new variables. We have these columns, 55,000 rows, and some number of we have 8 columns, 55,000 rows. We're thinking about replacing those 8 columns with maybe just 2 columns. Can we pick 2 variables from our 8, or can we make 2 new variables from our 8 original ones that represent most of the information that was in the full 8? But what we're going to get are actually new variables. The first 2 principal components, each of them is going to be of length 55,000, whatever the length, whatever the dimensionality here is. And the way I can get them is, so remember that with PCA, we're taking our matrix, M by N, for which there is a covariance matrix, N by N. And we are going to get these eigenvalues and eigenvectors of capital S. Then the first principal component, let's call it Y1, is equal to some linear combination of those columns. And the way I can write that, I could say X times, let's call the first linear combination, let's call it E. E1. If X is M by N, and E1 is the first eigenvector, E1 contains those coefficients, like 0.35 times column 1, plus 0.351 times column 2, plus 0.346 times column 3, plus dot, dot, dot. Those coefficients, these coefficients, are in here. So if I do this matrix multiplication, this will do that linear combination I want. Column 1 times that, plus column 2 times that, plus column 3 times that, plus dot, dot, dot. So this is going to give me a new variable. M 55,000-ish numbers. I can do the same thing for the second principal component, like that. Let's compute these, and then I'll show you what they look like. So PC1, let's call it Y1. You can actually do this in one fell swoop if I just say the PCs are data times eigenS vectors, which is the same thing as PCA dollar rotation. Same thing. So I have 8 columns here. I only want the first 2. Each column is a principal component. The first principal component is in the first column. If I were to do, okay, so here's the output from the eigen decomposition of the covariance matrix. What is the variance of the first column of these PCs? What is the variance of the first principal component? It's on the screen. Yes. The variance of the first principal component is 30.69 because that's the first eigenvalue. Are there going to be any columns that have a bigger variance than that? There can't be, because this is a linear combination of the columns that was chosen so that that number is as big as possible. So if I say, give me the variances of each of the columns of my original data, so column by column, give me the variances, it's not possible for any of these to be as big as this. This is the most informative way to linearly combine all your variables. Any one variable is a linear combination. It's 1 times that variable plus 0 times all the other ones. So that you know that this is more informative than any of these individually, or at least as informative. Can't be less informative. So the variance of this new variable, 55,000 numbers, is that right there. And if I wanted to answer the question in the exam, I would want to plot the first 2 principal components against each other. So let's do PC1 versus PC2. That's what I was asking for. What I got a lot of the time was a plot of these things. These coefficients. So I saw this a lot. PCA dollar rotation 1 to 2. The left plot, that's taken all of your all of your 54,000 genes, for which you have 8 observations each. Taking all of that huge amount of data and boiling it down from 8 dimensions to just 2. This is the most informative 2-dimensional representation of your huge matrix of numbers. So I need to have a number for every single record or gene in this case. I need to have 54,000 numbers. I've taken my 54,000 genes that were 8-dimensional originally and crammed them into 2 dimensions. So this is not an informative picture, or not a standard picture that you would make. If you wanted to look at the coefficients of the principal components, I would recommend just printing them off to the screen like this. But if you do this, you can inspect this for groupings, clusterings, Like if we saw, here's some points down here that kind of clump together, and then here's some points up here that kind of clump together. That would be indicative of there is structure to the data, because I can separate out these data in terms of this one variable. If there are individual points that are way off on their lonesome in one of these pictures, that's an indicator that those points are outliers, potentially. In 8 dimensions, if you look at it in a 2-dimensional way, they look different. So in some way, they are distinct from the others. That's what I was looking for in this picture. This picture, and in terms of comments, I didn't grade strictly on comments. The only comments I could come up with this picture were a couple. One, I don't see grouping. I don't see distinct blobs or regions. I don't see outliers that are very obvious. I'm not very worried about outlying values in this picture. There are some points that, obviously are further away than most of the others. But the fact that they are kind of gradually getting thinner and thinner as you move vertically, as opposed to everybody's right here, huge gap of space, and then here's one point. I'd be more concerned about that being possibly an outlier than this picture. The other thing that I see from this picture is that the first principal component, 30, has a variance of 30.7. And do you guys remember approximately what proportion of the total variance that was? What proportion of variance did the first PC explain? It's a lot. That's a big number compared to the rest of these. Here's another kind of... Here? Okay. So when we do principal components, the one on the right, like this picture on the right? This picture on the right is a scatter plot of these 8 numbers, or these 16 numbers. Yeah, these 8 pairs. So there's 8 points on the picture. First one is at x equal that, y equal that. So these are just the numbers, the coefficients that go into creating the principal components. They are not the principal components themselves. So a principal component, think of it as a new variable. In our case, we're treating columns as the variables. So a variable is going to be 54,000 numbers in it. And it is defined as a linear combination of the original columns. So the variable has 54,000 numbers in it. The linear combination, in order to do the linear combination, I need these 8 numbers. These 8 numbers are not the principal component. They are the coefficients that are used to make the principal component. So they are interesting to look at because the coefficients themselves tell us about how the variables are contributing. Like in this case, they're all basically the same number, so that's saying it's an index kind of thing. It's an overall average of all the 8 variables. That does tell me something, but it does not tell me this type of thing. This picture right here tells me this is not about how the variables contribute. This is about the underlying structure of the data itself. I've taken my high-dimensional data and I've crammed it into a smaller package that I can make a picture of. In this case, I'm looking for groupings of the genes, outliers in the genes, yes. So we are treating it like, in this case, we're treating it like our rows are our observations. Our columns are our variables. So we are looking for structure among the columns in terms of the rows. We could turn it around and look for structure among the genes in terms of the columns. Okay, so this is a handy picture. It tells you, this is kind of maybe this is one of the things you would always do, I think, if you did principal components. So things I would always want to know if I did principal components: what proportion of variability is explained? Is it much? If it is much, there's structure. What are the coefficients that make up the important principal components? That tells me about what is the nature of the structure. And a picture like this helps me a little further, I guess, to understand the nature of the structure, but also to identify possible outliers and unusual features in the data. Okay, so one last thing on the exam. For the exam, okay, when we did PCA, we got this as the coefficients that make this the first principal component, and that looks exactly like what we've seen in other examples in the class, and in particular, it looks exactly like what we saw when we simulated some data. So I simulated some normally distributed data that had a common, between all the variables, there was a common correlation of a reasonably high number. So there was strong pairwise correlation. And we got something that looked exactly like this. So what that tells me is that these data are correlated. The columns are correlated. You can see that if you were to do correlation of data. This is the correlation matrix. Let me round this so it's a little bit easier to see. That's the correlation matrix. So for example, column 1 has correlation 0.99 with column 2. If we plotted the first two columns, they're going to look a whole lot alike. Highly correlated. So take one of these, or take one row of our data set, one gene, and consider what we are doing in the exam. In the exam, part C of number 3, you were told to do a 2-sample t-test comparing control assays to treatment assays. So for gene 1, do a hypothesis test. Is there evidence of statistically significant difference in the means comparing control and treatment? So you could do that t.test, data first row. Let's go get all the control observations. Those were in column 1, 3, 5, and 7. And let's compare that to gene 1, all the treatment columns, 2, 4, 6, 8. Also, we were going to say var equal to false. So we can do a t-test, and I could pull out the p-value. P-value is 0.66. All right. When we did that just now, what issue is relevant? So what's the biggest issue that comes to your mind with doing a 2-sample t-test with those numbers? Anything come to your mind? What assumptions does the t-test make? Okay, so one of the assumptions of the t-test is normal distribution. We haven't even looked at that. I know that's going to be hard to check anyway because I only have 8 numbers. What else? What's another core assumption? For you guys who took 645 with me last semester, we started talking about regression. There were core assumptions of regression that we started with, but we ended up getting more flexible and saying we can do regression even when there's correlation if we handle it right. T-test, does it assume anything about independence versus correlation? Is correlation okay, no big deal, or what's the... Yes, so the t-test assumes independence. Most off-the-shelf statistical methods assume independence. So if I'm going to do a t-test on these 8 numbers, then this is a problem, because this is saying there's a lot, these are not independent of each other. They're highly correlated with each other. If they are not independent of each other, the assumptions of the t-test are not valid, which means your p-values may not be right. And you saw that in the exam. I didn't expect you to be able to diagnose all this. These were just like one of the first data sets that I found. I went to this geo repository for microarray data, or for a variety of omics types of data sets, and these were like the second or third entry, most recent entry in the database. You know, published in 2016. That's why I got them. So if you were to do that, pull out some data, it's got treatment and control. Let's go do a t-test. Without thinking carefully about what you're doing, you could get some weird results. So this was the histogram of the p-values. If you did it correctly, there were about 4 different versions of computing the p-values on the exam. The main distinction was we wanted a 2-sample test, and we needed to make sure we got the right columns for control and treatment. If you got everything lined up right, this was your histogram of p-values. And based on what we talked about in class, that's not a normal looking histogram of p-values. If there's nothing going on in the data, all null, this should be flat. If there is some subset of the features that are differentially expressed, it should be mostly flat, but then a peak of small p-values over at the left. But we have not seen any reason to justify a picture like this. Short story is, this is correlation. Correlation ruined our inference in this case. We did an incorrect approach to statistical inference because these data are so correlated. They are kind of paired data, and some people mentioned that in the exam. Shouldn't we be doing a paired t-test? And I think that's a good insight, because you have donor 1 and donor 2, and donor 1 gave you treatment and control cells. But certainly doing just a 2-sample t-test gave us an issue. So if you are doing omics analysis in your real world, and you compute p-values and you see something like this, something is wrong. I would not trust the results from this analysis. So if I did this analysis, I got these p-values, and then I turned around and got q-values or false discovery rates, I wouldn't believe those either. The q-values and false discovery rates depend on the p-values themselves having been computed correctly. And in this case, they were not, because this is not a valid shape for a p-value histogram. All right, so have a look at the solutions. Let me see if I've said anything more eloquent about that here. So on part 4 of the last problem, I was fishing for you to comment about this issue. So the principal component analysis suggested that the variables, the columns, were highly correlated. You can see that in the sample correlation matrix, and that causes issues. So that's a critique of using a t-test. A paired t-test might be better. Some people suggested a bootstrap. A bootstrap would be better. Okay. So ANOVA assumes... So ANOVA would assume the same things that t-test does. ANOVA and t-test are almost exactly the same thing. T-test is regression. ANOVA is regression. ANOVA assumes normality. It assumes independence. It assumes equal variances. There is a non-parametric version of ANOVA, though. So Kruskal-Wallis is a non-parametric ANOVA. So there are non-parametric techniques to several things, but ANOVA would be in the same boat as we are here. We could do regression that allows for correlated data, like we did in 645. But I definitely don't want to do something that assumes independence when you have data like this. So you've got a couple of things to think about whenever you are encounter omics data in practice. One thing to always keep in mind is, are these biological or technical replicates? These are definitely not biological replicates. We have 2 people giving us 8 columns of data. So that kind of feature to a design can really cause problems. Okay, what else did I want to do? I was going to talk a little bit more today about next-gen sequencing and start introducing us to the methods for doing statistical analysis with next-gen sequencing. So let's start. Under articles on eCampus, there's a paper that's written by Robinson and Huber from 2007. There's a Robinson 07 and there's a Robinson 08. And I wanted to talk about Robinson 07. So this came out, 2007, and there's a handful of methods that do similar things for analyzing next-gen sequencing data. This was one of the earlier methods that was proposed for RNA-seq, primarily data, but also applicable to other types of next-gen sequencing applications. We've talked about this issue when we're dealing with next-gen sequencing data, this issue that the response variable is a count. When we're playing with microarrays and some other technologies, the response variable is like a fluorescent intensity, which is a pixel intensity, a light intensity, and that's basically a continuous number. It varies so much that it could be treated basically as a continuous number. So t-tests and things like that are perfectly fine. Next-gen sequencing, though, we've talked about the fact that we get these counts. We get read counts. For each of thousands of genes, I saw 322, reads that overlapped with that gene, or 81, or something like that. And they're all counts. And the fact that they're counts, while that may not seem like a big deal in and of itself, the assumptions of the methods that you use to do statistics are important. And kind of like this issue, correlation breaks the t-test. If I have data that are not normally distributed, that can break the t-test as well. And read counts are not normally distributed. They're more like Poisson distributed. But it's more complicated than Poisson even, because of what in the statistical literature is called overdispersion. So if you think about a Poisson random variable, that is the outcome of some process, a random process, where events are happening at a particular rate. There's X number of phone calls to the 911 processing center in Houston per hour on average, for example. Something that's unique to the Poisson distribution is that its mean is equal to its variance. So Poisson is nice because it is applicable to these counting kinds of circumstances. And so it's natural to bring that in as a model for those types of circumstances. But just because you have count data doesn't mean you have Poisson data. And in particular, it's not uncommon in the real world to have count data that don't really follow a Poisson distribution. One way that can happen is for there to be Poisson-like counts, but they have more variance than the model, the probability model allows for. Poisson says variance equals the mean. It's possible for variance to be bigger than the mean. And when that happens, it's called overdispersion. So this whole paper, and then another paper by Anders and Huber, which is also on eCampus, talk about the same problem, which is how do you do testing? T-tests don't work with these data because of this count issue. So how do you do it? How do you do statistical inference, differential expression analysis with count data? Right. And the way that it is done in both cases, this paper right here, Robinson et al., is the source of this stuff in the NGS.R script that is called edge-R. So if I scroll down in NGS.R to line 187, there's this blurb about edge-R. So edge-R is a package in Bioconductor, and it implements the Robinson et al. Stuff. Down below edge-R, we'll go through this with an example together when we can, probably next time. Down below edge-R is DE-seq2. And DE-seq2, same idea. Let's do Poisson inference, but it's over-dispersed. It was just developed by a different set of authors, and it's got some slightly different techniques to it. The normalization stuff that it does is a little different. The algorithm is a little bit different. But the first one, the earliest one, was edge-R by Robinson et al. So I wanted to at least show you the paper. Robinson et al. So let's look at the model. What are we saying here? Okay, here's how they formulate the model. Yij is a count. That's the response for class I. That just means comparison group I. No, actually, that means, yeah, that means comparison group I, like treatment versus control. It would be two-class case, treatment versus control, but more generally there could be however many. And we're going to refer to the columns as libraries. So if you do next-gen sequencing, or if you work with people who do that and hand you their data, you'll hear this language of a library. That means we took tissue sample from this mouse, we extracted DNA or RNA, we ran it through the next-gen sequencing instrument, and at the end of that, we have our huge pile of reads. And those reads have been aligned to a reference genome. That constitutes the library. So it is the collection of all the reads for mouse 2, say. In other words, Yij is the ith row, jth column of our M by N matrix of read counts. So here is the key assumption. We have what it makes sense to think of, at least initially as a starting point. We have Poisson data, because they're counts. But we're not going to say that they are Poisson because of this over-dispersion issue. So I'm going to show you next time over in eCampus under Notes is just a page of algebra. Where did it go? Oh. A page of algebra. Why is this? There we go. Why it won't show me the whole screen. I'll show you this next time, I guess. But basically what's in this file that is called negative binom is working out, if you start with a Poisson distribution and it's not, or there's over dispersion, so the variance is not equal to the mean, you can work out that what you end up with instead of Poisson is a different distribution. So what both of these methods use, edgeR and DESeq2, what they do is they say y, the read count, yij, is Poisson with some mean parameter, call it lambda I, and then it says that lambda I has its own distribution, alpha beta. That's called a hierarchical model. That means usually we would say like my response is normally distributed with mean mu. Mu is some parameter in the population that we need to estimate. It's a fixed number that I just don't know. What we've done here is something like that. We said our response has some probability distribution. That probability distribution has a parameter. But then what we did was said that parameter has its own distribution. So really we're saying that y given lambda is Poisson, and it turns out that if you obtain the marginal distribution, this is a conditional distribution, if you obtain the marginal distribution of y, it is negative binomial. So maybe you've heard of negative binomial, maybe not. It's just another distribution, and it corresponds to a Poisson distribution that's over-dispersed. So this is appropriate for count data where there's over-dispersion. And if you do much with next-gen sequencing, you'll see this used a lot, negative binomial. It forms the basis of edgeR, negative binomial. It forms the basis of DESeq2. Right. So if you see negative binomial being bandied around, or negative binomial regression also, that's where this stuff comes from. We have count data, but it's over-dispersed. So the rest of the paper says, basically this is the main idea of doing inference with sequencing data. We're going to have one of these negative binomial distributions. Once you have a distribution, it's just a matter of estimating the parameters and doing tests on them. So the rest of the paper is trying to estimate mu and estimate this phi thing. If we can estimate those, we can put p-values on the genes. I think that's as far as I'll go in the paper itself. We're out of time. Next time we'll come back and we'll go into R, and we'll look at a couple of, or at least one example data set, and I'll show you what edgeR and DESeq2 do for you. It'll look a lot like what we've done already with t-tests, except instead of t-tests, we are going to use a negative binomial-based test instead of a t-test. Once we do this test, we want p-values, and then we're going to want false discovery rates, just like we've done with t-tests so far. So basically with next-gen sequencing, as long as you pick the right method at a bioconductor, a lot of what you'll end up doing is similar to what we've already done. You could do clustering, you could do PCA, you could do p-values and FDRs and all of that. Okay, questions about today's material? All right, I'll see you on Wednesday. Your homework, when is your homework due? Sunday. Okay. So we will have a Q&A this week, and I can help in that one. When was it assigned? Before spring break. If we need more time, we can do that. I think I said on the discussion board, why is this doing this? That the first problem can be extra credit. So there's a fair amount of R programming in the first problem. I think it's a good exercise, but if it's killing you by eating all your time to do it, you know, that's okay. But number 2 is a pretty straightforward problem. RNA-seq data and use DESeq2. So you can follow the ngs.r example to do this problem. All right, I will see you Wednesday. The Q&A takes place in 411, which if you walk all the way down and take a right at the very end, it's that last set of offices way at the end. 6 o'clock p.m. Wednesdays. Mm-hmm.